Hi everyone, I'm Rosemary from Molecular Cloud, and today we're interviewing MSP iGEM. So please tell us more about your team and your project working with the Oak Processionary Caterpillar.
Hey, first of all, thank you for having us. And we're from Maastricht in the Netherlands. Our program is called, most of us are studying, we're all studying at Maastricht University, but most of us are studying at the Maastricht Science Program, which is science college in English, and it's interdisciplinary, so we are free to choose our courses as we like. And then we also have one master's student who studies systems biology, and we have one first year biomedical sciences student. But we're generally a very international team, so we're from many different countries, and actually only have a few actual Dutch people in it. So we we didn't actually couldn't work in the lab till September because of COVID and everything, the restrictions. But so we can actually work with the OPC, but we did work on bacteria, and we are actually assembling our fighting strain, so to say, currently.
So in the beginning, for the summer, we mostly focused on dry lab work. So we created a network model, which would model our silencing RNAs and the effect of the silencing RNAs on the organism. And that is how we...
Maybe you want to start explaining the project, actually.
Oh yeah, okay. So we are tackling a more local problem, or it's a local problem right now, and it's called the Oak Processionary Caterpillar, which is a pest. It's a little caterpillar that attacks especially oaks, and the natural habitat of those is more towards the south, for example, Italy. But unfortunately, it turned into an invasive species because climate change made everything go a little bit warmer, so the caterpillar was like, yeah, more ecosystem to live in. So it wandered up and is now almost found in all European countries. And since it has no predators or predators in those regions, it can populate the areas very quickly. And that is very, very bad for the ecosystem because it goes out of balance. The trees are stripped bare to their bark, so all the leaves are eaten, and then the trees can be befallen by other diseases and are weakened. And another problem with this caterpillar is that it has these toxic hairs. And these toxic hairs, if they touch your skin, you get rashes. If you breathe them in, you get respiratory issues. And that is for humans, it's for animals. And these hairs, they're teeny tiny, and they can break off, and they can fly over 500 meters. They can also swim on water. So they spread very, very easily. And especially when it comes to the shedding period, when they start to prepare themselves, they shed all of the hairs, and that time is very, very dangerous. And therefore, some methods are used to get rid of the caterpillar, but these methods also attack other caterpillars, and they are bad for the ecosystem themselves. And so we decided, hey, we can find a better solution, better than these original pesticides, something that does not damage anything else in the ecosystem and only attacks the caterpillar. And I'm not sure how far familiar you are with the central dogma, but from DNA it goes to RNA, and then it goes to proteins. And if we want to interfere with the RNA so that there are no longer proteins produced and the cells can no longer survive, and that is how we want to control the population of the caterpillar. So we are using RNA interference, and our bacterium will produce teeny tiny silencing RNAs, and these silencing RNAs are taken up by the caterpillar, and then the caterpillar hopefully ingests them, and the cells in the caterpillar will die, and the caterpillar will also no longer be able to survive. What is special about this method is, since the silencing RNAs are very specific for the Oak Processionary Caterpillar genome, it will not attack any other caterpillars, any other animals, any other plants. So very much in the beginning, we started to do a blast search to see our silencing RNAs that we designed, would they attack anything else that is in the environment? And we spent quite some time to make sure it is only specific for the Oak Processionary Caterpillar. And with this method, we can restore the balance in our ecosystem here in the Netherlands, because if we do not start fighting the caterpillar right now, it will spread even further and it will become a problem in all of Europe. And with today's travel, it might even become a global problem. And so that is our main idea. So while we're not able to go into the lab in the summer, we worked on this network model where we modeled how our silencing RNA would actually have an effect in the caterpillar's body, in the organism. And we identified that 4 targets would have a really good effect, or like a bad effect, and would enable us to fight the caterpillar. Furthermore, we are working also right now on a spread model so that we can determine if there was a population last year and how big it was, where will it probably be in the next year or the year after, so that we already know where we can preventively spray our pesticide, so that we do not need to look always like into the trees, hey, this tree needs to be sprayed, and we don't need to go around, but we can specifically say this area is very endangered, let's go there, specifically look for it, for example. And yeah, we also focused a lot on human practices, obviously. We had a survey to ask people, hey, what do you think about a project, about GMOs in general? Because that is a big concern, because we are planning, or we would like to release these bacteria that produces silencing RNAs, because putting the bacteria on the leaves is much more efficient than actually just putting the RNAs on the leaves, because the bacteria would protect the RNAs and then secrete them. And so more RNA would come onto the leaves, and it would stay longer for almost 10 days, which is exactly the time period that we need for it to work. Well,
And it would be cheaper.
Yes,
well.
it would obviously be cheaper. And the second option, if we could not get a permit to use live bacteria, we could potentially keep the bacteria in the lab and make them self-cracking so that they release all the RNAs, the silencing RNAs, could then take the silencing RNAs, turn them into a spray, and could only spray the silencing RNAs. The problem with that is they will stay, depending on the weather, and be active for only 3 to maybe 7 days, which means we need to spray it 2 to 3 times, which is a lot less effective and a lot more expensive. And we want to bring also down the costs, because the pesticides that are used right now are also very costly. So yeah, that's what we are working on, and we are finally able to go to the lab, and so we are now working on actively creating our bacterium.
I see. I think this is a very important cost, especially because you said that the hairs of the bacteria spread so far and can even travel in water. It seems like it can be even such a harm to people as well, and as far as being an irritant and respiratory irritant as well. So have you thought about, has your team thought about what would be an example of a mass delivery method? So for example, if you use the spray with the bacteria, how would you go about spreading vast forests of oak trees to ensure delivery? Or are you hoping that there is some, or are you aiming to have some sort of epigenetic effect so these silencing RNAs can be inherited into future caterpillars?
Well, first of all, that's a very interesting approach, but yeah, it is also like a rather difficult, and especially from like a safety point of view. So we prefer to just spray them onto the trees, and our team has actually already contacted several people that work for the municipality here in our city that actually like exterminate the caterpillars, because they are like such a big health concern that there are people really employed full-time just to take care of them, and they already use a biological pesticide, a BT toxin, which they actually spray on the trees with modified tractors. So it is really just a normal tractor with a kind of contraption on the back, that moves 45 degrees to either side and sprays like a fine mist of these bacteria. And then they drive around the streets, basically, and then they spray both sides of the streets where there are trees, and everything is covered. So we hope to use the same method, because we're also using bacteria, so it should work the exact same. And
That's great.
yeah, also we did not plan large scale spraying that well. You could of course spray it with like an airplane, but there's problems with the heat that is generated there, that it might damage the bacteria. But generally that is also like a big aspect to our project, because the bacteria pesticide they're using right now has, like we already said, it has like a lot of effects on other insects as well, especially other caterpillars. So they cannot spray that in big forests, because in city ecosystems, well, there aren't many caterpillars around, and like even if they all die, it doesn't have a great impact on anything. But of course, in big forests, the problem of the caterpillar is even worse, because there are many oak trees that it can affect, and it can basically be a breeding ground for them. But they just cannot use their current pesticides because they're not specific enough, and the damage to the ecosystem would be even greater. So yeah, we really hope to also change this part.
Okay. It's great that your municipalities already have something implemented for the delivery of the pesticides. I think that would be awesome, to deliver the spray locally as well. So you discussed about your modeling that you've been using to it before getting into the lab. Can you tell us more about what your modeling system looks like?
Unfortunately, the person that did most of the modeling is not here. So I don't think we can tell you too much about it. I could look something up right now while you ask a different question, if you're interested in that, and see what he wrote down so that we can have a look at that.
No problem.
Basically, we created a metabolic network model that was for Drosophila, actually, because it was the closest organism that we could find related to the Oak Processionary Caterpillar. So we couldn't find any expression data for the Oak Processionary or related species. But we used the Drosophila model to model the impact the knockdown of the genes would have. And we couldn't use that for all of the genes, because some genes are specific to the Oak Processionary. But we found out that 4 of the target genes we would have a significant impact, at least in Drosophila. So we transferred this to the OPC as well.
Okay, thank you, Eva. I think that's a great system, especially using multiple targets, using what you can from Drosophila, but, adapting that to the Oak Processionary Caterpillar. I think that was a great adaptive system to create. So you mentioned that you were also working with human practices, and you created surveys to see how people in your communities would feel about introducing a GMO. So what was the feedback like?
Eva, do you want to answer that? You worked with Ronja a little bit.
Ronja is actually here,
yes.
so she can tell you something about our survey.
Yeah, so we conducted quite an extensive survey on the general attitudes and also the knowledge about the caterpillar and how it's dealt with. So we asked about like personal experiences of the people, if they have encountered the caterpillar, and also what they think about like GMOs and synthetic pesticides, and what would their reaction to our approach be. So the survey data, it shows quite clearly, it was actually very nice that the caterpillars are very close to human habitation, and they have a very sometimes very radical like effects to the humans living there, not only the humans but also the pets, and so on. In general, people are quite reserved on using GMOs and bacteria and methods like these to fight like anything. But our survey data shows that when we ask the question of using GMOs and these pesticides in the context of fighting the caterpillar, people were a lot more positive, like significantly more positive towards these methods. And they also thought that our method would be more effective in comparison to the current methods that are used. So I think this summarizes the data quite nicely.
Definitely, I think it's great that the public is more open to using the GMOs, especially as a pesticide, especially when pesticides have this, negative stigma or being full of harmful chemicals and being harmful to, the environment as well. Using a bacteria is definitely a much safer alternative, especially when it's targeting just that one caterpillar species.
Yes, and we have also tried to approach this stigma issue and everything like this with our YouTube channel, and for example, making these educational videos, and for example, we had this radio interview with the student radio Maastricht where we talk about the project. So we are trying to spread awareness both on the issue itself and also how we could solve it.
Awesome. Those are really great and helpful and informative initiatives, and I'm sure they will, have a good educational cascade and the perception of GMOs. So is there anything else your team would like the audience to know about your project?
Could you repeat?
Are there any other details or initiatives that you would like our audience to know about your project?
Oh, I would like to mention our journal initiative. So we are making this big journal of different iGEM projects, and we also have the this Muckle journal version of it. So it's basically the same articles, but simplified more towards the general public. So not only scientists or people with scientific background can read about these cool projects, but also the normal people too.
That's really awesome. I think that's great that your team is getting involved in science communication and science education and making it more easier to interpret and translate and having it more easily accepted. That's wonderful. Well, MSP iGEM, thank you so much for meeting with me today. Your team has an incredibly innovative project, and I'm sure it's going to, the idea is definitely very helpful to the ecosystem, and definitely, how the pest affects people as well in the community. And again, thank you for meeting with me today.
Yeah,
Thank you
very much for having us.
Good luck in the iGEM
Thank
Jamboree.
you for having us.
No problem.
Thank you. Have a
Our
good
pleasure.
Bye.
Take care. Bye-bye.